The Electron Beam Accelerator Facility is a new laboratory for basic nuclear physics research. At CBAF, scientists can advance our understanding of matter itself by probing the quarks and gluons in the atom's nucleus with beams of electrons from a superconducting accelerator. CBAF has an educational mission as well. The CBAF Science Series features scientists and engineers who are gifted at making their work not only informative, but fun. The Science Series is held once a month during the school year in the CBAF Center Auditorium. The presentation is live, spontaneous, and for the most part, unrehearsed.
Good evening. Welcome to the October Science Series of 1995. It's my pleasure tonight to introduce our speaker, who is Professor Charles F. Stevens of the Salk Institute in San Diego, California. Professor Stevens grew up moving from place to place because his father was in business and got transferred a lot, and also because he was in the Navy. He attended college at Harvard University and majored in psychology because his freshman year that was the most interesting course he took. While he was in college, he got some opportunities to do research projects, and this was pretty exciting to him. And so when he finished, he realized he actually wanted to go further into science, and he chose to enroll in medical school at Yale. After getting his MD, again with a little more experience in research, he realized he wanted to really spend his life doing research and not, I guess, curing people. So he has had professorships at the University of Washington in Seattle, at Yale University Medical School, and now he's at the Salk Institute. He's spent many years doing research on memory, nerves, the brain, cells, transmission of information. And it's my pleasure tonight to introduce him to talk about a topic which I just can't remember. Oh yeah, it's how memory works. Join me in welcoming him. Thanks a lot.
Thank you, Bev. Okay, this is an informal talk, and so if you have questions, you should stick your hand up. And if I don't see you, I can't see very well because of the lights, you should yell out. Okay? And if you do it too much, then I'll mention that. but really, I don't think you'll do it. But it would really be good if you ask questions, because this is a level of audience. I usually talk to people who have PhDs. I don't usually talk to high school students, and so I may not do it quite right. So it'd be good for you to yell out and tell me stuff. Okay, people who come into neurobiology really come in with big questions. They come in with questions like, how do we think? Why do we fall in love? Why are some people smarter than other people? How do we remember things? What are emotions? And many times you can't make much progress on those questions. But recently, we've been making a lot of progress on the question of how memories are stored in the brain. Not the question of exactly what happens in the brain when you're getting a memory. And so what I'm going to tell you about tonight is some implications, some things about how memories are stored, what happens inside your head when a memory is stored. And I'm also going to tell you some interesting other implications of this mechanism, because there are things that happen when memories go wrong that are medically interesting. So I'm going to tell you about both of those things. The first thing I have to do, though, is I have to tell you how the brain is organized on the inside. I have to tell you some facts about what's inside your head. Inside your head, there are a lot of special cells called nerve cells. Okay, or also neurons. And a nerve cell all cells have a cell body. And then coming out from that cell body, like branches out of a tree, are a lot of tubes coming out in all directions. And these tubes are called dendrites. The dendrites are where signals from other nerve cells are transferred to this nerve cell. So here's the cell body. And then there's a little wire. It's not really a wire, it's a tube, but it's like a wire that comes out of this and it goes over to other nerve cells. And that wire is called an axon. Okay, so let's get an idea of how big these things are. Here is a human hair. This is a cross-section through a human hair. Okay, and this is about how big a nerve cell would be on a cross-section of a human hair, if you pulled one out. How long is this axon here? And axons are very different lengths. In you, the longest axons are ones that start here in your back and run down to your foot. So it's a single tube, and it's this big compared to a human hair. So there could be thousands of them in the diameter of a human hair, and they are about this long. And the longest one I know of in the world record, is the ones that start at the base of a giraffe's head. One single cell, one tiny long extension of a single cell that runs all the way down from the giraffe's neck, 14 feet, down to the giraffe's stomach where the diaphragm is. So they're really long. When I was in high school, I was wrong about a lot of things. I could give you a really long list, but one of the things I was wrong about was what nerves do. If I prick my finger with a pin, then everybody knows that the signals are going up nerves all the way up to my head. And I had the idea that there was a chain of nerves. And I wasn't quite sure how big each one was. I thought maybe there was one that was about this long and then another one. And I thought that there was a chain of these cells that started here and went on up. That's totally wrong. One of these axons starts up in your brain, and it goes all the way down to your arm, and it comes down here to the end, and it has special little endings on it. So when you touch there, you generate nerve impulses, and they go back up. So there's a single cell that runs all the way down to there, and that's what the nerves are made of. Now, inside your brain, there are a lot of these nerve cells, and this is one of them. And here's another one I'm drawing down here. I'm going to make it a different color, actually. Since I have more colors here, I might as well use them. There's the cell body. These are the dendrites. They go up like that. Here's the axon going out. And where this cell here, where the black cell goes down and comes near the red cell, there is a special contact that the black cell makes onto the red cell, and I'm going to draw a better picture of this in a second. But this special contact is called a synapse. Okay, I'm sorry for all the terminology, but you really have to be able to have some words to talk about these things. So this is the cell body of a nerve cell. Messages are sent from the black cell to the red cell, and they're sent and they get transferred from the black cell to the red cell at this special place, which is called a synapse. And I'm going to tell you a little bit more about how synapses work in just a second. Just to give you an idea of how complex the brain is, if you imagine a piece of brain the size of a grain of rice, okay, just an uncooked grain of rice, inside a volume of brain the size of a grain of rice, there would be 10 million, I'm sorry, there would be 1 million neurons, 1 million of these. There would be 10 billion synapses, and there would be 20 miles of axons. So there, inside a grain of rice, there are 1 million cells like that. There are 10 billion contacts between those cells, and those 10 billion contacts are made by 20 miles of these axons. And you have total in your brain that many nerve cells, and you have total in your brain that many synapses. So there are a lot of them up there, and it's really complicated. Okay, now I'm going to talk a little bit about how information goes from this cell to that cell, because it happens by a funny way. And it turns out that it's important, the way that it happens. Okay, so here is the axon. They gave me lots of these, so I'm going to use another one and do it right. Okay, here is the axon from the black cell from before. And it's actually like a very fine tube. And there's a swelling on it right there. Is that okay? I think it just sort of fell down. How's that? Is that okay? Okay. Here's the axon, and there's a swelling. This is the black cell from before, and this is an axon from the black cell, and here is a dendrite from the blue cell, and it has a little thing that sticks out like that. Okay, so what I've done is I've taken, oops, I got the colors wrong. No, you can't get everything right. Okay, I'm taking this part right here, and I'm making it really big. So this right here, this black thing right there, is this made big, and this dendrite here, the red dendrite, is turned blue now, and it's right there. Okay, and inside this, and this is the axon, and this is the dendrite. And inside the axon, there are a bunch of little balls, and those balls are filled with a chemical. And the particular chemical that is in most of these little balloon-shaped things in your brain is called glutamate. And there are a bunch of special proteins that are embedded in this membrane that makes the outlines of this dendrite. When a nerve impulse comes down here, and a nerve impulse is an electrical signal, and people know all about them, but I'm not going to tell you about them unless people ask for some reason. When the nerve impulse comes down, it activates some special machinery in this axon, and that special machinery makes the membrane that's around this vesicle here. These things are called vesicles, synaptic vesicles. Let me write it down. These little balls here are called synaptic vesicles, and they're full of chemicals like glutamate. And I'm going to draw it big down here. So here is a synaptic vesicle, and here is the membrane. So I'm taking this part here and I'm making it big, and here is another one. And what's happened is that one of these synaptic vesicles has bumped onto this membrane, and there's some special proteins that do this, and it has fused onto the membrane, so what was inside right there has now come out. So this is a little ball filled with glutamate, and about 10,000 molecules of this glutamate, which is an amino acid. It comes out, and it diffuses from there to there. And now the cell is going to turn red again. This is getting confusing. Okay, so we're looking at this here, and we're looking at this part down there. Okay, and match them up. And so this is this part made really big. And the glutamate diffuses across like this, and it binds onto special proteins here that are called receptors. And when it binds onto those receptors, it makes the receptor change its shape, makes a hole in it, and some ions go through, and that's an electrical signal. Okay, there are two reasons why you should be interested in this. The first reason you should be interested in is that almost all of the medications that are used for, that affect the brain, and in fact, almost all of the drugs of abuse work by changing synaptic transmission. So, for example, barbiturates that are used for sleeping pills modify synaptic transmission at a certain class of synapses. Coffee, the reason that coffee keeps you awake is that it modifies the synaptic transmission at other synapses, caffeine. Valium works at a class of synapses. Ethanol, alcohol, works at another class of synapses. In fact, I don't know a single drug that affects behavior and affects the brain that doesn't work by working at synapses. And they work different ways. Some of these drugs mimic the neurotransmitter, and so they're sort of phony neurotransmitters that you eat, and they come and they bind onto these receptors. Other ones block the neurotransmitters. So, for example, it's an amazing thing to think of, but when you move, anytime you move, when I'm talking, you move muscles. When you move those muscles, every time you move a muscle, your nerve is sending a message down to synapses that axons make onto the muscles. They're releasing a chemical. The chemical down at muscles is glutamate, is acetylcholine. That's the name of the chemical at muscle synapses. And the acetylcholine goes from the nerve cell over to the muscle cell. It opens up these little holes and it makes the muscle contract, and that's how you move your arm. And if you ever read a James Bond mystery or something like that, they're always killing people with these toxins. And if you get bitten by a cobra, for example, the way the cobra kills you is it has special chemicals in it that go on, and they bind on to the receptors, and they keep the acetylcholine from binding onto its binding site and making the receptors work. And so cobra toxins kill you that way. The Indians in South America catch animals by poisoning them with curare-tipped arrows. Those work by blocking receptors. When you if you go and have surgery, they give you what are called muscle relaxants. Those muscle relaxants work by blocking the receptors like that and stopping the transmission between, in this case, nerve and muscle rather than nerve and nerve. And they're quite specific, so you can get ones that only affect muscles and don't affect your head. Okay, so the first reason then you should be potentially interested in how synapses work is because most of the drugs that affect us, antidepressants, antipsychotic agents, anesthetics, sleeping pills, all the drugs of abuse, alcohol, anything you can think of, all work at synapses. The second reason why you should be interested in synapses is because synapses are the site of memories. That's how memories get stored. You have billions and billions of synapses in your head, and when you store a memory, what you're doing is you're making some of those synapses stronger and you're making some of those synapses weaker. And I'm going to tell you a little bit about how that works.
One question.
Yes, please.
How much information gets stored at each synapses?
The question was, he was asking how much information gets stored at each synapse. Well, that's a hard question to answer, you know, because the synapse has different grades of strength, and so it's really hard to know, you know, how much. It's hard to answer how much, hard to quantify it. But for one memory, there are millions of synapses involved. So only a small fraction of anything you would recognize as a memory. Okay, so the way scientists work on this these days is the following. You get a mouse, the mouse gives you his brain, you shred the brain up, and you put it in a culture dish in an incubator, and the individual brain cells in there try to grow a brain, and you can look in and see them. And so you can record the electrical signals from individual nerve cells. You can record one and the other one. So you can study them exactly and precisely. And if you do that, here is the axon. This is the axon. My color coding is long since gone. And here is the dendrite. These red things are the synaptic vesicles that are full of glutamate, and here are the proteins in the dendritic membrane. This whole thing is the synapse. And just to review it quickly, I know I just said it, but let me just say it again. Nerve impulse comes down here. Synaptic vesicles fuse with the red membrane, release the glutamate on the inside. Glutamate diffuses across, binds onto those green molecules there, makes them have a change that generates an electric current by ways I'm not telling you. You can't understand it because I didn't tell you, but it's not that hard to understand. In this cell, that electric current flows down here, and that's a signal for this cell, which it then uses. Okay. Now what I'm going to do is I have some special little wires that I can stick over here, and I can generate a nerve impulse over here anytime I want to. And I have other little wires that I can stick in here, and I can record a nerve impulse anytime I want to. And I'm going to make a graph here of time during my experiment. So from here to here is 1 hour. And this is the size of the response in this cell here. The nerve impulses have the special property that they're all the same size. You don't have big nerve impulses and small impulses, nerve impulses. You either have one or you don't. So I cause nerve impulses to come here. Every time one comes, all that business happens sending the transmitter across. I record over here, and I measure the size of the response, which is a little current. And that's kind of complicated how it flows, but it's not essentially difficult. It's just I didn't explain it to you. There's a little current over here, and I measure that. And I plot the size of that here. And I plot it along like this, and it stays constant for a long time. And then I do something that I'm going to tell you in a second, but I haven't told you yet. And after I do that, then every time I stimulate this response, every time I stimulate, so I'm recording from here, every time I see how effective this synapse is at causing a response over there, what I find out is it's bigger. And it stays bigger. Here it stayed bigger for an hour. There are experiments where it stays bigger for 2 weeks. So this synapse now is a more effective synapse than it used to be. There's another thing that I can do right here. If I do that other thing, if I use this synapse in a particular way, it gets weaker. And it stays weaker for hours and hours, virtually indefinitely. And, and this getting stronger and getting weaker is one of the keys in the storage of memories, and how we know that I'm going to tell you in a couple of minutes. Okay, I did something here. Or I did something different there, and we're just going to talk about the first thing, the thing I did here to make it stronger. This getting stronger has a special initials. It's called LTP, and that's not important. It stands for long-term potentiation. It's just this is the way people talk about it, and it means that the synapse is stronger long-term. Okay, it's gotten bigger like that. What did I do here to make it get bigger? What I did was I used the synapse lots of times, but I used it together with a bunch of other synapses on this same cell. I'm not going to tell you, it's known, but I'm not going to tell you how it works. It's not a secret, it just takes too long to do it. But these proteins here are very special proteins, and they have a very tricky way of being able to detect when glutamate was released by this cell and simultaneously when other cells, other synapses were active. They're special kind of coincidence detectors, and if this synapse is active at the same time that other synapses are active, these proteins can sense that, and they let something happen. And I'm going to tell you what they let happen, but it's just so we have a way of talking about it. It's not going to change your understanding of things that much. What they let happen is they let calcium ions come into the cell. And those calcium ions that come into the cell trigger a whole bunch of biochemistry, and that biochemistry is what makes the synapse get stronger from then on. And it gets stronger because every time after that happens, some signals go back and forth between these two cells, and then more neurotransmitter, more of this glutamate gets released each time it comes by. Okay, so basically then, what memories are the pattern of strengthening and this weakening of synapses in complicated patterns all through your brain. And the thing that triggers that is the simultaneous activity of this synapse plus other synapses together. Okay, so I'm going to do two things now. The first thing I'm going to do is I'm going to tell you how we know this is related to memory. And the second thing I'm going to do is I'm going to tell you some other implications of this, some other things that this same memory-like process is good for. Okay, the cells that I put in the culture dish here, the rat gave me his brain, I shredded it up, I put the cells, they try to grow their own brain in there, I can study them. I can also study those cells in the living animal. And those cells come from a particular part of the brain called the hippocampus. And the hippocampus is, everybody has one, they're in there, and rats have them, all the animals have them, and it's just a special part of the brain. And the first person, first person to understand how important the hippocampus was is a woman named Brenda Milner. And there was a man named H.M. who around 1960 had epilepsy, and he had intractable epilepsy. He had seizures over and over and over again. And for a reason that I'll tell you later, having uncontrolled seizures is not good for you. A few don't hurt, but over and over and over again definitely isn't good for you. And that's why there are a number of people in this room who will have seizures during their lifetime, and a few more or less doesn't matter. But repeated seizures do matter, and that's one of the reasons why neurologists always like to make sure that seizures are controlled so people don't have them. This person had uncontrolled seizures. One of the things that doctors do, and they're much better at it now than they were in 1960, is this: usually the seizures start in a particular part of the brain. You have a lot up there, you can spare quite a bit, so they just cut out the part, and you can't even tell that they've done it. In the old days, they were a little cruder, and they used to cut out more of the brain, and they weren't quite so good at it. They didn't have the good techniques that they have now for mapping out what the brain is doing. Now, when they do epilepsy surgery on someone, they map out the brain, and they make sure that they stimulate parts of the brain, and they find out where the language areas are, and they make sure they don't get those. There's certain areas of the brain that are specific for language, and you don't want to take those out because life without language isn't worth living. And so they avoid those and take out neighboring parts. In the old days, they didn't do that. And so this guy, H.M., they took out parts of his brain on both sides because he had epilepsy on both sides. And the part of his brain that they took out was the part that contained the hippocampus. After the surgery was over, the very first time Brenda Milner came to study him, she came to talk to him because he could talk fine, he was intelligent, but he couldn't remember things very well. And so she came and she talked to him, and he could answer questions. He was a very intelligent person, but he couldn't remember anything. He couldn't remember what he had for breakfast. He used to look at the same comic books over and over again and laugh the same times every places because he couldn't remember ever having read those things before. And if you think about it, he's still alive. He's in Boston. He lives in a hospital there, and he's a very nice man, but he has to be hospitalized because if you think about it, if you couldn't remember anything, you couldn't enjoy anything. You couldn't enjoy watching a television program because you couldn't remember it. Stand-up comics is is all you could watch, because you couldn't remember the plot of a movie. You couldn't remember the plot of even a sitcom. You couldn't remember anything, and that would be terrible. So this guy, H.M., Brendan Milner discovered, had lost his hippocampus, and then he couldn't commit any new memories at all. He remembered everything that happened perfectly before the operation. He also could learn motor skills. So he could learn new manipulations. So for example, if he hadn't learned how to play tennis, he could learn how to play tennis and get quite good at hitting the ball, but he couldn't remember the rules for the game. Yes? I'm sorry, I can't hear. Can someone else hear? Because I can't see and I can't hear.
How could he remember good enough to read if he couldn't remember anything?
He couldn't remember anything new, but he remembered everything old. He remembered everything that happened up until the time of his operation, and he had learned to read before his operation. So if you don't have a hippocampus, you can't remember things. There's some other studies that show that memories are stored there temporarily for about 2 weeks, and then during that 2 weeks they get put other places in the brain. So if you don't have them, you can't store them for that 2 weeks, and you can't put them other places. But you can remember everything from before, absolutely everything, perfectly. Better than we can, in fact, because he doesn't learn anything else that interferes with it. Okay, so thank you for asking the question. Good for you. So they figured out from HM that this was definitely an important part of memory, and so they started doing animal experiments on that. And so now you can do animal experiments, and you can show that the hippocampus is required for memory in these animals. But how do we know that it's long-term potentiation, this strengthening of synapses that's available, that is what stores the memory? And the way you know that is several. One of the things is that there are some drugs that you can take, that you can give to animals, also some you can take, that block the action of, I'm going to draw this special protein here with a green, with a blue circle around it. Okay, this is the special protein that lets the calcium ions come in. There are certain drugs that you can give to animals that block that particular protein from working, and that protein is, remember, that's the thing that detects that this synapse is active and other synapses is active, detects both are active simultaneously, and then triggers the changes that cause long-term potentiation. Quite a bit's known about that, but I don't have time to tell you about it. Okay, so if you give a drug that blocks this, animals act like they don't have a hippocampus. And you simultaneously block LTP. So if you give one of those drugs, after the drug was given, if you did the same manipulation that would cause LTP before, the size of the response would stay unchanged.
Are there people born without a hippocampus?
No. I don't know, maybe, but they would have, oh, I'm sorry, the question was, thank you, Bill. The question was, are the people born without a hippocampus? And that is, the hippocampus is such a deep part of the brain that people who are born without one would not have, they would have so many other problems that you wouldn't notice. The hippocampus would be the least of their problems. Yeah. Okay, so if you give these drugs, then you block LTP, and you also block learning and memory. Just, yes, please.
But once you give those drugs to block the calcium,
yeah,
you're coming back down to normal.
Yeah. Well, no. Yes. Okay. I understand the question.
Because with the calcium, we went up to LTP.
Yeah.
Because I want to find out how would we regulate it down to.
Right. If you let's say that you give this drug over here. What would happen then? Nothing. You have to give the drug over here before you do the thing that causes LTP. It only blocks the triggering of it. It doesn't block it once it's there.
Right. Because calcium is not a normal flow in that system.
That's right. And calcium would not normally come in.
Right.
Right. Absolutely.
Okay.
Yeah.
So you get that through the acquisition of drugs. Or is there a normal function in our?
Normally this blue protein lets the calcium come in, and that calcium is critical for triggering the memory. That's what triggers it. Okay. If the calcium doesn't come in from the outside inside the cell, then the memory doesn't get triggered. And if you give that drug, then it blocks that protein from working. It doesn't let the calcium come in. Okay, I wasn't quite clear about that. just incidentally, I'm not in any way I'm not here preaching against drugs or anything like that, but just sort of out of interest. The street drug called angel dust, whose scientific name is phencyclidine, actually works by, one of the things that it does is it blocks that thing, and in experimental animals you can block memories by giving them angel dust or phencyclidine. That's one of the things that will block the function of that blue receptor.
I understand there are drugs that improve memory. How do they work?
There are drugs that people will, the question is, there are drugs that improve memory, how do they work? And that's the question. The answer is, there are drugs that people will sell you that they say improves memory. And in very certain special circumstances, they will improve memory in a way that you can measure in a special circumstance, but they don't work any better than coffee. Yeah. Oh, I'm sorry, excuse me, I couldn't see you very well. Yes, please.
Through hypnosis, they say that memory is, like, you can recall memories from early childhood and all kinds of things. How does hypnosis work? I mean, how does...
Oh, okay. Well, the question is how hypnosis works and how you can recall early memories. That's actually an interesting question. And you know, let me. My ambition is to get through before 8 o'clock, because I know there are people who want to go. But it's a very interesting question, because there have been a whole lot of things recently about early memories that have been recovered, and there have been lawsuits about that. There have been like child abuse cases where the daughter suddenly remembered that her father and some psychiatrist helped her recover these early memories. And if you ask me about that at the end, I'll tell you about it. Okay, but let me go ahead with this. Okay, so how do we know this is the process that is underlying memory? And the chain of argument goes like this. First of all, we know the hippocampus is where these memories are. And the second thing we know is that this LTP happens in the hippocampus. We know that if we have a drug that blocks this LTP by blocking this blue receptor there, that also blocks LTP and it blocks memory in animals. And more recently there have been, now there's a technology where if you tell me a particular protein in a mouse's body, not yours, in a mouse's body, I can make a mouse that doesn't have that protein. And the mouse may or may not live. It may be perfectly normal, but it's missing. It's a mutant mouse that misses just one protein. And my laboratory and other laboratories have studied these mice, and among the proteins that are missing are some that are involved, after this calcium comes in, they're involved in triggering the LTP. And so you have mice that are missing a particular protein later on down the stream, one I'm not telling you about. This synapse works perfectly well. There's nothing you can tell about the synapse that isn't working well. But those mice don't have any LTP, and they can't get new memories. They can't get spatial memories for like the kind they have with their hippocampus used. So there's a chain of reasoning here that links up LTP, which you can study in a culture dish, with the LTP that you can study in the whole animal with learning and memory experiments. And so this is how we know that LTP at synapses is vital for learning and memory. Okay, now let me go back and talk about two other things. And one is strokes, and the other is development. Okay, what is a stroke? Those of us who do not die of heart attacks will likely die of either cancer or strokes. I think strokes kill, heart attacks kill most of us, and the ones that don't get killed by heart attacks mostly get killed by strokes, and the ones left over die by cancer or cars. Okay, so you didn't know this was going to be such a cheery talk, did you? Yeah. Okay, so how do strokes kill you? What a stroke is it's like a heart attack except up in your brain. A blood vessel, most strokes, a blood vessel from arteriosclerosis gets plugged up, and the blood doesn't go to a part of your brain, and because the blood doesn't go there, the nerve cells die there. Now, if you have a stroke, and I'm going to draw Okay, this sheet of plastic now is the surface of my brain, or here's the surface of my brain. And I have a stroke, and that stroke supplies blood to that area of cells there. And you have so much brain up there that you really can, any little bit of it you can get by without. Big bits you can't get by without, but any little bit you can usually get by without. And a stroke would stop the blood supply going to this blue spot right there. Okay, so this is the stroke area. I'm going to call it the stroke core. Now, what happens with a stroke is that the cells in the stroke core die, but the cells around the stroke also die. So a big area here of cells dies, not just the ones that are in the stroke core, but ones in the neighboring region. And the reason they do is because of a phenomenon called excitotoxicity. What happens is that when cells here start dying, they release chemicals, and those chemicals make the cells around fire lots of nerve impulses, and those nerve impulses make the cells around them fire more nerve impulses, and there's a big region around the stroke core where all the nerve cells are firing like crazy because of the stroke in the very middle. And cells that are excited too much kill themselves. They excite themselves to death, and this is what excitotoxicity is. And excitotoxicity basically is the same mechanism as you have for forming memories. Remember that I said that, Remember I said that when you trigger a memory, what happens is that calcium comes in the cell here, and a little bit of calcium is absolutely vital for triggering a memory. But remember that I also said that this synapse tries to form memories when this synapse is active at the same time all the other synapses are active. Around a stroke, every synapse is active, and so they're all simultaneously active, and so they're all trying to store memories, and they're all letting calcium ions come in. And the calcium ions that come in at all of those places start flooding the cell with calcium. And a little bit of calcium is absolutely vital. It's necessary for triggering memories. But there are mechanisms inside of cells that keep the calcium at very low concentrations. When the calcium gets to too high concentrations, it activates a bunch of enzymes that start eating the cell up and just kill it. And so the mechanism of excitotoxicity is this extra calcium that comes in. And that extra calcium that comes in all the synapses at once it starts out excitotoxicity, and the treatment for excitotoxicity are drugs that can block this calcium that comes in here excessively. And there are, in England right now, there's a drug in clinical trials for stroke patients, and also the same thing happens with head trauma, like people who fall off bicycles and things. There's an experimental drug that blocks this blue protein there that keeps the calcium from coming in when it's overexcited. And in animal experiments, it decreases the size of the stroke damage down to a very small part. Okay, so one of the things that's interesting about the synaptic transmission memory mechanism is that, like all of pathophysiology, all of the things that go wrong with you are basically the same things that go right, but in exaggerated form, and in an exaggerated form they're bad for you. And so a lot of the damage in strokes is basically your brain trying to memorize things when there's nothing to memorize and there are too many cells trying to do it. Okay, during development, there's an interesting thing that happens. When our genes tell our brains how to wire up. So all of those synapses are all made by the right wires, basically, but they're not quite right. And there is a modification procedure that where the connections between cells get modified. Some connections get stronger, some connections get taken completely away, and that happens by virtue of experience. And this can be demonstrated in experimental animals, and so different people, identical twins, can have different brains, anatomically, physically different brains, on the basis of their experience. Now, this is something that many of you can appreciate intuitively, and that is that, let me say one more thing. During a particular age of our life, the brain is modifying itself, tuning up its circuits so it does things the best way, and then different parts of the brain at different times stop that modification. They freeze in the wiring that's there, and they don't change it anymore. And so for certain things in the visual system, where your visual system has to tune itself up, if you have children with very bad vision, if their vision isn't corrected by a certain time, glasses don't help anymore, and the reason is because their brain got wired wrong, so it can't do the best kind of precision analysis of images that it could otherwise. And so there's a particular window of opportunity during which the brain needs experience in order to form itself properly, and something that you may all have had experience with, even if you don't know it, is language ability. And that is that up until the age of most of the people in this audience, you can learn to speak a foreign language perfectly, and you can learn to speak it without an accent. There's nothing I could do to learn a foreign language without an accent. And in fact, by the time I was 22, there was nothing I could do to learn a foreign language without an accent, because the language circuits that have to do, any human in the world can learn any language perfectly with no accent if they do it before they're about 17, 18, something like that. If you wait until after you're 17 or 18, nobody can learn any language without an accent, because the parts of the brain that have to do with processing language are plastic up until that time, and then they close down. And so there are these various windows of opportunity that we have during development where it's terribly important that people have the experiences that they have, because the brain is wiring itself up. How does it do that? It does the same thing right here, that the brain, during the early part of development, the same mechanisms that are used for storing memories are also used for deciding which synapses are going to be made permanent and which ones are going to be removed. So most of you in this room have more synapses than you need in places you don't need them, and you're removing them still. And so as you remove those synapses, then you can remove them, you can put them back again. Everything is quite plastic up there until a certain period of time, after which it doesn't happen anymore. And the trigger for removing or stabilizing synapses is this calcium influx through this blue receptor there. Okay, so what I've tried to do then is I've tried to give you a quick picture of how scientists think about memory these days, of how they study it, what we think memories are, and what some of the implications are of the mechanisms for some other things in health and disease. Thank you very much.
Some of you may want to ask questions now, so if people are leaving, please leave quietly.
Okay.
Read the question.
Let me let people leave for a second. I'll answer it in just a second.
Let people leave. We're going to leave. Just please leave quickly, and the people who are going to stay.
What's your name is?
Yes, Chuck Stevens.
Thanks. You did a good job.
Oh, thank you. See ya.
Is it possible that memory could be altered?
I'm going to answer questions, yeah.
If you'll sit down there.
Okay, no,
Okay.
I don't. I need to get going.
Oh, okay. Be altered, how do you mean?
Like, if you were to go to a psychiatrist, you know, and like
Yes.
they were to supposedly look back in your past, would it be possible for them to